This will be a quick review of DNA, how it can store information and be duplicated. We've learned in previous lessons about the cell cycle. All cells have a birth date, so they come from pre-existing cells. They grow, and if they're going to go on to reproduce, they're going to need to duplicate their chromosomes in preparation for mitosis. Mitosis is the process where the duplicated chromosomes will be delivered to the daughter cells. Now, we've learned that chromosomes are composed of DNA molecules wrapped around proteins. It's the DNA molecule that is the hereditary molecule. It is the molecule that has the capacity to store information, to be duplicated, and to change over time. Here we see the DNA molecule. It is a double helix, or a twisted ladder. Two strands are sort of coiled around each other. We have the rungs of the DNA ladder made of 4 kinds of chemicals. The abbreviations are A, T, C, and G. And you'll notice a pattern in the pairing. The G chemical always pairs with the C chemical, and the T chemical pairs with the A chemical. So the rungs of the ladder are made of those 2 kinds of pairings. Importantly, there's a weak chemical bond in the middle of those rungs here, so that is going to permit the molecule to be duplicated. The DNA double helix will unwind, and then the 2 strands will separate. When the 2 DNA strands separate, it'll look something like this. Now here we have, in this part of the diagram, we still have the double helix structure of the single DNA molecule. Now the strands have been separated here. Now there's a whole team of proteins involved in DNA duplication. One of those proteins is going to untwist the DNA molecule. Other proteins will help stabilize the single strands. But the star of the show is going to be DNA polymerase. This is the one that's going to construct the new strands of DNA. When the old strands are separated, they are templates to build the missing strand. Because of the pairing rules, then letters can find their sticky partners. And then along comes DNA polymerase that will chug along the template strand, linking together the new letters, linking the backbone together to form a stable new strand of DNA. It'll also work on the other strand. So the result of this process is going to be the duplication of one DNA molecule into 2 identical DNA molecules. And that looks like this over here. So if you check for a moment, these 2 double-stranded DNA molecules are identical. Now, as it happens, DNA polymerase is a pretty cool protein. It also has a proofreading function. So as it's building the new strand of DNA, it's checking and correcting mismatched letters. And the result is a pretty impressive error rate. 1 mistake every billion letters copied. In this diagram to the left here, we see a duplicated DNA molecule. However, duplication has not been fully completed. So the 2 identical DNA molecules here, so one here and one here, are still attached at this location. So the end result of DNA duplication along the length of a chromosome looks like this. Now, we've also learned that the histone proteins are proteins that DNA wraps around. When it comes time to enter mitosis, then, the first stage of mitosis, in the condensing stage, the DNA is going to tightly coil up around these histone proteins, and then coil again, and then the coiled sections will supercoil, and you'll get a chromosome, as shown over here. So this is our cartoon chromosome. So notice the chromosome looks like an X, because they are duplicated chromosomes but still attached. The chromosome was duplicated because the DNA molecule itself was duplicated. However, an unfinished section here keeps them attached. Now, mitosis then is going to be the process whereby these duplicated chromosomes will be segregated into daughter cells. So here we are now in mitosis, the 4 stages of mitosis: condensing, lineup, separation, and division. And again, this is the small blue wedge of the cell cycle. So during interphase, chromosomes are duplicated, which means DNA was duplicated. Then in the first stage of mitosis, the chromosomes will begin to coil up. They condense, and so they appear as X's. Duplicated chromosomes but still attached. The nuclear membrane breaks down. This allows microtubules to search for and find the chromosomes, lining up the duplicated chromosomes, and then the duplicates detach, and one set of chromosomes goes to one side of the cell, the other set goes to the other side. These are identical chromosomes now. They were duplicated during interphase, and then the cell will divide into 2 cells. The nuclear membrane will reform, and then the DNA will begin to unwind from the histone proteins so that its information can be accessed. So mitosis is the way single-celled organisms like amoeba and paramecia can reproduce. Again, the DNA in the nucleus must be duplicated, and then mitosis will deliver full sets of chromosomes to each daughter cell. So in a sense, mitosis involves moving the duplicated DNA to make nuclei in two daughter cells. In the next part, we'll see what's happening during interphase. The cell will be using the DNA to make proteins.